Welcome back, dear students. Today I'm going to be talking about blood typing. Blood transfusions in humans were a risky procedure until the discovery of the major human blood groups by Karl Landsteiner, an Austrian biologist and physician. Until that point, physicians didn't understand that death sometimes followed blood transfusions when the type of donor blood infused into the patient was incompatible with the patient's own blood. Blood groups are determined by the presence or absence of specific marker molecules on the plasma membranes of erythrocytes. With their discovery, it became possible for the first time to match patient-donor blood types and prevent transfusion reactions and death. Antigens, antibodies, and transfusion reactions. Antigens are substances that the body doesn't recognize as belonging to the self and that therefore trigger a defensive response from the leukocytes of the immune system. Antigens are generally large proteins but may include other classes of organic molecules including carbohydrates, lipids, and nucleic acids. Following an infusion of incompatible blood, erythrocytes with foreign antigens appear in the bloodstream and trigger an immune response. Proteins called antibodies, immunoglobulins, which are produced by certain B lymphocytes called plasma cells, attach to the antigens on the plasma membranes of the infused erythrocytes and cause them to adhere to one another. Because the arms of the Y-shaped antibodies attach randomly to more than one non-self erythrocyte surface, they form clumps of erythrocytes. This process is called agglutination. More than 50 antigens have been identified on erythrocyte membranes, but the most significant are classified in two groups: the ABO blood group and the RH blood group. The ABO blood group. Although the ABO blood group name consists of three letters, ABO blood typing designates the presence or absence of just two antigens, A and B. Both are glycoproteins. People whose erythrocytes have A antigens on their erythrocyte membrane surfaces are designated blood type A, and those whose erythrocytes have B antigens are blood type B. People can also have both A and B antigens on their erythrocytes, in which case they are blood type AB. People with neither A nor B antigens are designated blood type O. ABO blood types are genetically determined. Normally, the body must be exposed to a foreign antigen before an antibody can produce. This is not the case for the ABO blood group. Individuals with type A blood without any prior exposure to incompatible blood have performed antibodies to the B antigen circulating in their blood plasma. These antibodies, referred to as anti-B antibodies, will cause agglutination and hemolysis if they ever encounter erythrocytes with B antigens. Similarly, an individual with type B blood has preformed anti-A antibodies. Individuals with type A, B, Blood which has both antigens don't have preformed antibodies to either of these. People with type O blood lack antigens A and B on their erythrocytes, but both anti-A and anti-B antibodies circulate in their blood plasma. Rh blood groups. The Rh blood group is classified according to the presence or absence of a second erythrocyte antigen identified as Rh or rhesus. It was first discovered in a type of primate known as a rhesus macaque, which is often used in research because its blood is similar to that of humans. Although dozens of Rh antigens have been identified, only one, designated D, is clinically important. Those who have the Rhd antigen present on their erythrocytes, about 85% of humans are described as Rh positive, Rh plus, and those who lack it are Rh negative, Rh minus. Note that the Rh group is distinct from the ABO group, so any individual, no matter their ABO blood type, may have or lack this Rh antigen. When identifying a patient's blood type, the Rh group is designated by adding the word positive or negative to the ABO type. For example, A positive, A plus means ABO group, A blood with the Rh antigen present, and AB negative, AB minus means ABO group, AB blood without the Rh antigen. ABO transfusion protocols. To avoid transfusion reactions, it's best to transfuse only matching blood types. That's a type B plus recipient should ideally receive blood only from a type B plus donor, and so on. That said, in emergency situations, when acute hemorrhage threatens the patient's life, there may not be time for cross-matching to identify blood type. In these cases, blood from a universal donor, an individual with type O minus blood may be. Recall that type O erythrocytes don't display A or B antigens. Thus, anti-A or anti-B antibodies that might be circulating in the patient's blood plasma will not encounter any erythrocyte surface antigens on the donated blood and therefore will not be provoked into a response. One problem with this designation of universal donor is if the O- individual had prior exposure to Rh antigen. Rh antibodies may be present in the donated blood. This may cause problems for the recipient, but because the volume of blood transfused is much lower than the volume of the patient's own blood, the adverse effects of the relatively few infused plasma antibodies are typically limited. Rh factor also plays a role. If Rh individuals receiving blood have had prior exposure to Rh antigen, antibodies for this antigen may be present in the blood and trigger agglutination to some degree. Although it's always preferable to cross-match a patient's blood before transfusing in a true life threatening emergency situation. This is not always possible, and this procedure may be implemented. A patient with blood type AB plus is known as the universal recipient. This patient can't theoretically receive any type of blood because the patient's own blood having both A and B antigens on the erythrocyte surface doesn't produce anti-A or anti-B antibodies. In addition, an Rh plus patient can receive both Rh and Rh plus and Rh minus blood. However, keep in mind that the donor's blood will contain circulating antibodies, again with possible negative implications. That's it, dear students. Good luck. See you.